Imagine a laboratory that learns in the way a child does. It tries, it stumbles, it watches the result, then tries again, a little wiser, a little softer. The room hums with a steady, almost sleepy rhythm. Robotic arms move like careful hands, pipetting tiny droplets into tiny wells. Cameras watch. Sensors listen. An algorithm, not a person, guides the flow, sending the next suggestion into the air like a breath. The lab is not just following instructions. It is writing them, testing them, and listening for what the world quietly answers back. This is the dream of the self-learning lab, a place where artificial intelligence and automated instruments link together in a loop of curiosity. The model proposes an experiment. The machines run it. The results come back, and the model learns how to do better, over and over, like tides smoothing stones. It's delicate, methodical, and it could be astonishingly powerful. At bedtime, the mind can widen. Questions come gently forward when the day's noise fades. Could such a lab discover a cure faster than we've ever known? A new antibiotic for stubborn infections. A tailored therapy for a rare disease. A vaccine assembled like a poem, line by line, to match the shape of an unseen enemy. Could a self-learning lab watch nature's patterns, then place the right pieces together with the patience of a gardener? But in the same quiet, another thought arrives. If machines can explore the living chemistry of cells, if they can propose designs for molecules or proteins or genetic edits, could they also stumble toward something harmful? Something careless, or something no one intended? Could the same capability that helps us heal also carry risk? These are not questions to race through. They are questions to hold gently, like a fragile specimen glowing under a microscope. To understand what self-learning labs really are, and what they are not, we can step slowly through the science. What does it mean to teach a machine to design? What do these systems actually do, moment by moment? How does a lab translate a computer suggestion into a physical, wet world experiment? Where do the guardrails live, and how do they hold? Like any living thing, the story of science breathes. It widens with new tools, then settles into new habits. Tonight, we'll wander through the rooms of this emerging idea. We'll meet the software that imagines, the robots that carry out gentle procedures, and the human values that frame what is allowed and what is not. We'll listen to the old conversations, ethicists, physicians, biologists, who have long asked how to move forward without losing our way. And as we do, we'll hold the balance that matters most. Self-learning labs are not magic, and they are not doom. They are careful machines whose power depends on human intention, skill, and responsibility. Between the next cure and the next crisis lies design, policy, and the deep, patient art of care. Come closer. Listen to the soft whir of motors, the steady tick of a thermal cycler, the faint scent of ethanol, and clean glass. The lab is awake. The models are waiting. And the first truth to learn is perhaps the simplest. Intelligence, whether in minds or in machines, grows by asking good questions. What questions, exactly, are we teaching these labs to ask? We'll begin with the early whispers, how the idea formed, and how the smallest steps became the first confident walk. Long before anyone dreamed of a lab that could teach itself, scientists dreamed of a lab that could keep up. In the old days, and by old, we mean not long ago at all, researchers pipetted by hand, hour after hour, bending over plates, counting drops like beads. It was beautiful work and brittle. Each experiment took time and thumb skill. Repetition was demanding. Discovery moved like a cautious river. Automation entered as kindness. Liquid handlers learned to move with unerring precision, especially for tasks the human hand finds boring, the 100th pipette, the 1,000th. Plate readers scanned results with the steady attention of a metronome. High-throughput instruments made it possible to try many variations at once. Imagine a painter who can lay out not just one shade of blue, but hundreds, and see how each changes in sunrise or shade. Then came something quieter, almost a personality change in how the lab thought. The old way, design an experiment, run it, read the result, design the next. The new way, let an algorithm help choose the next step. The algorithm remembers, weighs uncertainty, and gravitates to the experiments that promise the most learning. This approach, a kind of patient conversation with the unknown, goes by humble names: optimization, feedback control, active learning. But its essence is simple. The lab asks, where should I look next to learn the most? And then it goes there. When artificial intelligence models grew better at spotting patterns, this loop became more elegant. A model could propose a new chemical structure likely to bind a target protein. A robot could synthesize close variants around that suggestion, test them, and feed the results back into the model. The next design would sharpen, narrow, refine. This is not wizardry. It's a thoughtful treadmill, designed not to tire, but to teach. We've seen glimpses of what that means. AI systems learn to predict the folding of proteins into their exact three-dimensional shapes, a task that once took months in the lab and years of human nuance. With those shapes, the mind's eye can place molecules like keys into locks. Models can sketch a possible key. Automated instruments can try to make it a physical thing. And with each success or failure, the sketchbook improves. The image of a self-driving lab emerged from this harmony between thinking software and careful machines. Some research groups set up experimental platforms that can run day and night, adjusting conditions based on what the measurements say. The system exploits the gentle logic of trial and error, not in leaps, but in the smallest, safest steps. Sometimes it's materials, seeking better batteries or catalysts. Sometimes it's chemistry, searching for reactions that make a complex molecule flow more easily. Sometimes it's biology, charting how cells respond to different sequences or shapes. If you picture a self-learning lab as a place of bright screens and clear liquids, you aren't wrong. But it is also a place where constraints are thick and protective. Biological work is bounded by layers of review and containment, by biosafety cabinets that whisper clean air over gloved hands, by rules about what can be handled and how. These are not afterthoughts. They are the spine, the non-negotiable rhythm that keeps the lab safe for people and for the world outside. The early pioneers of responsible science spoke to this long before algorithms could propose experiments. At a seaside meeting decades ago, biologists gathered to set norms for a powerful new technique. They asked what kinds of experiments should not be done at all, which should be done with special care, which would be safe with modest precautions. They chose caution first, until the unknown became known. That was not fear. It was love of life expressed as prudence. Those principles still hold. The arrival of an eye in the lab does not loosen them. It tightens them. A system that can explore many possibilities needs a fence around the garden, not to strangle it, but to shape it. The fences are both physical and conceptual. Approved organisms only, non-pathogenic strains. Screened sequences, logs of every action, strict oversight. Tired ideas retired. The point is not alarm. It is stewardship. So we wander the aisles in our mind, past racks of clear vials and the soft glow of incubators, and we ask the next question gently. If a lab can learn to find the best recipe for a therapeutic molecule, can it also learn something we do not want it to learn? What stands between curiosity and carelessness? The answers live in both engineering and ethics, in systems designed to say no, as thoughtfully as they say, try this. Let's meet the stories that show how we have navigated these edges before the eras, when we found steps forward without stepping into harm. Science grows through small stories that cast long shadows. Picture the California coast in winter light. Decades ago, scientists gathered there to talk about a new way to recombine DNA fragments of life, joined in combinations no one had seen. The ocean breathed behind them. They set voluntary rules, advocating containment levels and careful experiments. They asked for time to understand risks before running too far ahead. That meeting, remembered for its simple wisdom, became a touchstone. It showed that communities of researchers can choose restraint, even when curiosity is strong. Since then, cautious bravery has helped humanity achieve improbable things. A tiny virus that once haunted the world was coaxed into a corner through vaccination until it disappeared from natural circulation. Another inherited disease, once mysterious, yielded to a logic of hemoglobin and a simple medication's surprising power. None of these victories were instant. They were mosaics of experiments, ethics, checkpoints, and patience. The rise of machine learning slipped into this stream. In one era, algorithms helped predict which compounds might bind a protein. In the next, they learned to imagine new compounds outright. In parallel, robotized systems made it easier to test them. A few teens spoke of robot scientists not to replace humans, but to reflect a workflow where hypothesis, experiment, and revision could cycle without constant hands-on intervention. The robots didn't think in the way we do. They followed patterns. But from those patterns, useful knowledge emerged. In the world of proteins, an AI learned to predict shapes from sequences. Structures that once needed crystals and X-rays could be drawn up by code with startling accuracy. That shift did not cure a disease by itself, but it changed the pace of understanding. If you know a shape, you can imagine how a medicine might fit it more clearly. If you can update that shape quickly as new data arrives, you can track a moving target. We saw, too, how speed can be a blessing. When a new respiratory disease swept across the world, scientists used computational tools to identify targets, to design molecules that might train immune systems, and to optimize formulations. The machine's role was not to invent life, but to help us see patterns faster, to simulate, to filter options so that the wet lab could focus on what mattered most. Manufacturing and clinical testing still demanded care, time, and oversight. The path from idea to approved therapy remained long because it must be. At the same time, discussions about boundaries never paused. There are lines of research that raise deep questions work, that tweaks biological properties in ways that ask for even more caution. Globally, advisory bodies sift evidence, recommend safeguards, and sometimes recommend stopping certain directions when benefits are uncertain or risks are too large. DNA synthesis companies formed consortia to screen orders, comparing requested sequences against libraries of known hazards. Institutions established review boards, layered safety protocols, training, and inspections. This is all part of the quiet backbone that lets science proceed without racing toward a cliff. Self-learning labs sit within this wider culture. Their strengths are real. They can search a large landscape of possibilities, not randomly, but with a compass that points toward learning. They can save time, reduce cost, and help discover new materials, catalysts, and perhaps medicines. Yet they are not unbounded. A model can propose a protein, but making it, testing it, and understanding its effects require careful steps, many of which cannot be automated away. Life is intricate. Cells are not beakers. They respond to context, and context cannot be fully scripted by code. One of the most important truths is this: designing harm is not a mirror of designing healing. The properties that make a pathogen dangerous are complex, tied to host biology, environment, and evolution. They are not merely sequences or shapes. They are the sum of many traits interacting, often unpredictably. The more deeply you look, the more you find that control is hard and lucky accidents are rare. For all their pattern-finding grace, today's models are not oracles. What they do well is accelerate the reasonable, not conjure the impossible. Still, the responsibility remains. We can admire the elegance of a system that learns, and at the same time, insist on rules that keep its curiosity safe. We can build labs that never touch certain kinds of work, that filter suggestions through layers of screening, that require human review and sign-off. We can use secure environments so that models trained for healing cannot be repurposed for harm. We can track every action in tamper-evident logs. We can design incentives, funding, publication norms, industry standards that reward safety, transparency, and care. Consider a different image now. A forest at dusk. The trees stand in quiet, yet underneath, a network of roots and mycelium trade signals and nutrients. It looks peaceful because it is governed by relationships. Self-learning labs can be like this, fleets of small decisions nourishing a larger aim, connected by rules and reviews that keep the whole system healthy. As we step toward brighter possibilities, new diagnostics found in hours, bespoke therapies tuned to an individual's biology, enzymes that clean rivers, we bring the same lantern with us. Cautious optimism. The question is no longer only can we build a lab that learns? It is, how do we teach it what to learn and what never to touch? The next part of our story travels into that teaching, into the metaphors and mechanisms that shape machines toward healing. Think of a self-learning lab as a garden that learns its gardener. You plant a seed, and the soil keeps notes. When you water in the morning, the leaves brighten, and the soil marks it. When you overwater in the evening, the soil remembers that too. Over time, the garden nudges you more of this, less of that, until it thrives within a rhythm you share. Inside real labs, the nudging is mathematical, but the spirit is the same. The AI proposes an experiment, not because it knows the final answer, but because it knows which question will be most informative next. It balances exploration and caution, surfing uncertainty the way a careful walker tests a stepping stone. In chemistry, this might mean choosing temperatures and solvents that are safe and likely to work. In biology, it means focusing on allowed, screened components, benign strains, approved constructs, and assays that reveal insight without risk. For listeners who enjoy metaphors, here's a useful one. A self-learning lab is like a choir. The AI is a conductor holding the score. The instruments, robots, sensors, incubators are musicians playing parts. But the sheet music includes rests. the conductor must honor them. In science, the rests are constraints, safety protocols, ethical guidelines, lists of what not to attempt, access controls, and the sober signatures of review. A good conductor doesn't chafe at the rests. They make the music coherent. In practical terms, that means layering safeguards from the first keystroke. Models trained for therapeutic design can incorporate filters that stop them from generating disallowed suggestions. The hardware layer can include interlocks and audits. If a protocol falls outside pre-approved templates, the system cannot run. DNA orders requested by any system, human or AI, are screened against databases maintained by specialized groups. Cloud platforms that host AI models can require additional review for sensitive domains, with activity logs that are monitored for anomalies. None of this is the exciting part of the story. And yet, it is the part that makes the rest of the story worth telling. There is another quiet lesson from the history of engineering. Objectives shape behavior. If you tell a system to optimize only for speed, it will. But it may cut corners you didn't intend. If you teach it to optimize for healing within explicit guardrails, to maximize patient benefit, minimize side effects, respect safety constraints, it will make different choices. This is why the human part of a self-learning lab is not the robot arm or the AI code, but the values we choose to encode: transparency, traceability, restraint. Technical humility is a virtue here. Biology is not a rigid machine. Genotype does not map to phenotype like a dictionary mapped to a word. The effect of a change often depends on context, cell type, environment, time. That complexity is a safety feature of the world itself. It means that even brilliant models must pass through screens, tests, and careful observation before any claim is trusted. It also means that deliberately crafting dangerous traits is not a straightforward endeavor. The world resists simplistic designs, and responsible communities make sure the path stays difficult. Meanwhile, the positive horizon is broad. A self-learning lab can search for molecules that blunt antibiotic resistance by disarming bacteria. Tricks not by escalation, but by cleverness. It can design enzymes that break down pollutants we once thought intractable, cleaning water like a reed bed does along a riverbank. It can help create diagnostic tests that respond quickly and affordably, bringing care closer to communities that need it. It can explore personalized medicine by adjusting formulations to a person's biology, as a tailor adjusts fabric gently around a shoulder. Equity matters too. Tools that accelerate discovery must not deepen divides. There is a future in which low-cost, well-governed platforms allow smaller labs and clinics to run safe, approved assays tuned to their patients' needs. Open standards can make collaboration easier without exposing sensitive capabilities. International agreements can harmonize screening and oversight so that safety is not a patchwork but a quilt. If we pull back even farther, we see a pattern that is strangely comforting. The immune system learns by exposure and caution, probing and remembering, guided by boundaries that keep its power from turning inward. It is a self-learning lab within us, ancient and elegant. It works well when it listens, when it keeps good records of what to attack and what to leave alone. Perhaps our external tools should imitate that wisdom. Learn quickly, remember clearly, and stand down where harm would otherwise happen. We can end this part with a soft truth. The question, could a self-learning lab design the next pandemic, is understandable, but it flattens a rich world. The more precise question is, how do we ensure that systems designed for healing are embedded in cultures, infrastructures, and rules that prevent misuse. That is the work already underway. Not a single gate, but a series of them. Not a single guardian, but many. Scientists, ethicists, policymakers, companies, and the quiet daily diligence of technicians who notice when something looks wrong and say so. The next breath in our story is about meaning. If we keep the garden safe, what does it give back? How will it feel to live in a world where cures are designed faster, where we respond to outbreaks with quiet competence, where we heal without haste? What does that do to our evenings, our families, our sense that the future is not a cliff but a meadow? Imagine that future evening. The day fades. The window shows a thin stripe of orange, then purple, then a blue that invites rest. In a hospital lab, a small machine hums. It learned from thousands of experiments run in shared, carefully governed facilities. It proposes a tailored therapy for a patient whose disease once baffled doctors. A clinician reads the suggestion, checks the safety flags, and approves a formulation proven safe in prior trials. The patient rests. Behind that quiet moment stands an orchestra of rules, sensors, and models aligned to one simple aim: reduced suffering. This is the best version of our self-learning labs, not a fevered race, but a long, calm stride. These labs are teachers and students at once. They teach us where to look. We teach them where not to step. They remember outcomes so we don't repeat old mistakes. They magnify our empathy by turning it into design. More effective drugs with fewer side effects. Diagnostics that return answers in hours instead of days. Materials that store clean energy and keep hospitals running smoothly. There will be questions we cannot rush. How do we keep the benefits global, so that care is not a luxury of one region, but a shared inheritance? How do we ensure that when the models change, they change in ways we can understand? How do we keep human judgment at the center, so that decisions carry responsibility and meaning? These are governance questions as much as technical ones. Supporting thoughtful policy, transparent auditing, and international cooperation is part of the science now. It always was. If a self-learning lab is a mirror, it reflects what we pointed at. If we set it to find faster pathways to vaccines in a crisis, we must establish processes that include community trust, clear communication, and ethical review. If we set it to design small molecules for neglected diseases, we must ensure that those molecules are affordable and available. If we harness it for environmental health, we must measure not only what we remove from water, but who gains access to the clean streams that follow. In this gentler future, response to new health threats is neither panic nor denial. It is practice. Simulations run in the background, so we know in advance which interventions are safe to test and in what order. Local labs run approved assays with clear boundaries. Orders for biological materials are screened, and logs are watched, not out of mistrust, but out of care. When something unusual appears, it is checked thoughtfully, not shouted about. The rhythm is slow enough to be sustainable, fast enough to be effective. And perhaps the deepest comfort comes from remembering that technology rarely outruns the wisdom we bring to it for long. We set the terms. We choose the objectives. We build the fences and the gates, and we train ourselves to notice when a latch is ajar. The awe we feel at what these systems can do, imagine shapes, refine recipes, steady our hands, does not have to be tinged with dread. It can be accompanied by the quiet knowledge that we have prepared together to guide them. So, Could a self-learning lab design the next pandemic? In responsible hands, with layered safeguards, the answer bends toward no. The incentives, the rules, the access controls, and the deep complexity of biology itself keep that door closed. Could these labs help design the next cure? Yes, by helping us learn faster, test safely, and translate knowledge into healing without losing our calm. The same loop that could race ahead can be tuned to walk with purpose. Maybe tonight, as you close your eyes, you'll hear the soft hum of a lab in your imagination. Not ominous, not heroic, just steady, pipettes moving, data settling like snow. A machine proposing one more gentle question, and a human curious, careful, kind answering with a nod. Thank you for sharing this quiet journey. If these bedtime explorations of science bring you calm and wonder, you are warmly invited to subscribe and drift with us again. We'll keep walking gently, listening for the science that helps us care for one another.